setup is? I think you can identify from the apparatus that this is Ganong's photometer. Can you identify?
Yes, ma'am. Yes, ma'am.
Okay, let me make it more clear. This is the Ganong's photometer, all right? And it's a doubly bent glass tube. And you already know the description. It has a wider end and a narrower end. And the narrower end and the horizontal part of the photometer is fitted with a capillary tube inside. And the narrow end is dipped inside a beaker containing colored water, which I have done. I have used blue colored water over here. And I have inserted a leafy shoot at the other end. And I've filled this entire apparatus with water. And yesterday, actually, I had introduced the air bubble. And today it has moved, it has gone that side. So I think showing you the air bubble over here, it's extremely difficult, but you can see the colored water rising into the capillary tube. Is it visible to you? The colored water?
Yes, ma'am. Yes, ma'am.
Yes, ma'am.
Can you see that? The colored water, it has gone up into the capillary tube and it has reached, you know, till here. Can you see that? All of you?
Yes,
Yes. Yes. So what has happened? Huh? Yeah. Can you all see? The colored water rising of the colored water into the capillary tube of a Ganong's photometer. Is it visible to all of you?
Yes, ma'am.
Yes, ma'am.
Yeah, very good. So what is the purpose of Ganong's photometer? A Ganong's photometer measures the rate of transpiration. It is not demonstrating, children. It is measuring the rate of transpiration. All right. And how does it measure? In order to start off this experiment, we have to first introduce an air bubble. And the air bubble, you know, ensure that only one air bubble is introduced. So you are going to set this entire thing up and expose it to sunlight for a few hours, which I did yesterday. All right. And after I expose it to sunlight, we will see that the air bubble moves towards the horizontal end, gradually moves up towards the horizontal end. And in the horizontal end, actually, there is a graduated scale where you can time the movement of the air bubble over a fixed distance on the scale. And that's how we find the rate of transpiration. You know, rate means we are trying to find out the speed of movement of the air bubble, and that we do it by calculating distance by time. Okay, when we time the movement of the air bubble over a fixed distance on the scale, that means for the first 1 centimeter, it takes how much time. For the second 1 centimeter, it takes how much time. Then the total distance by total time will give us the rate of transpiration. That's how it happens. So this is showing a Ganong's photometer. Now, why does the colored water rise? This might be another question. Why does the colored water rise into the capillary tube? The colored water rises into the capillary tube because the leafy shoot over here, it has transpired. Now, since it has transpired over here, it has absorbed water. Now, because of that, because of this transpiration, a suction force is being created. And because of that suction force, this colored water rises from the beaker into the capillary tube. So the associated questions with this particular experiment, first question they'll ask you, identify the apparatus. So you are writing it is Ganong's photometer. You are not writing only photometer because there are other photometers as well, like Farmer's photometer, like Darwin's photometer. So you are writing this is Ganong's photometer. That's the first thing. Then second thing is they'll ask you what is the role of the air bubble in this experiment. So you will write the air bubble helps us to measure the rate of transpiration. And how do we do so? We time the movement of the air bubble over a fixed distance on the scale, and that's how we find out the rate of transpiration. Now there's a reservoir over here, and the reservoir is operated by a stopcock over here. What will happen? Suppose the air bubble has already crossed this horizontal bar, graduated bar, and we know that the introduction of the air bubble into this apparatus is an extremely difficult job. So we don't want to introduce another one, but we got to use the same air bubble. So what we do, since it has already reached the end of the scale, we just open the stopcock and push the air bubble, and the water will flow down, and the water, the water is going to push the air bubble to the initial position, and then we can restart the experiment. So the question will be, what's the work of the reservoir? You will say that it pushes the air bubble to its initial position in order to restart the experiment. Then why have you taken colored water in the beaker? So you will write, colored water has been taken because the leafy shoot is going to transpire, because of which a suction force will be developed, and then the colored water is going to rise into the capillary tube. So this proves that the plant has transpired. This is going to prove that the transpiration is taking place, and because of which the colored water has risen up into the capillary tube. Any questions? Any questions, children? No, from this experiment? No. All of you are now clear with this experiment, because this is an important question which usually comes. All right. So shall I remove this?
Excuse me, ma'am.
Yes, ma'am.
Yes.
Ma'am, is all the water which is absorbed by the plant transpired? So that we are able to say that it's measuring the rate?
Yes, exactly, Divyanshu. We say that rate of water transpired is equivalent to the rate of water absorbed. We say that. But when we are writing what is the function of this device, what is this device used for, you are not going to write that it measures the water uptake, but it measures the rate of transpiration. Because, like we know that water transpired is water absorbed. Why do we say so? Because out of the total water absorbed, only 1 to 2% of the water is utilized by the plant for its different physiological processes. The rest, 98 to 98% of water is transpired. And that's why we say that the water absorbed is water transpired. Hence, we are, it's given in the book that it is measuring the rate of transpiration, which is equivalent to the rate of water absorbed. Am I clear, Divyanshu?
Yes, ma'am. Yes, ma'am.
Okay. So I'm removing this then?
Yes, ma'am. Yes,
okay. Now look at this setup. Can you see the setup?
Yes, ma'am.
Yeah. What does this experiment demonstrate? It shows that oxygen is evolved during photosynthesis. And actually oxygen is getting evolved, but I don't know whether you can see the evolution of bubbles or not, but I think you can see the bubbles. Yes, just a moment. You can see the bubbles, But my network is okay. I think you can hear me.
Yes, ma'am.
All of you?
Yes, ma'am.
Now is it all right?
Yes, ma'am. Yes, ma'am.
Yes.
Okay. Okay. I don't know whether you can see the bubbles, evolving and going up into the test tube, but you can see the bubbles over here. Can you see the bubbles, almost inside the water, which is contained by the funnel? Can you see the bubbles inside?
Yes, ma'am.
I don't know whether it's possible for you to see. Huh?
No, ma'am.
I'm not clear.
Ma'am, you can click a picture.
Okay, just a moment. There's a pinch of sodium bicarbonate I'm adding, that is baking soda. I couldn't get hold of the chemical, so I used baking soda, which is nothing but sodium bicarbonate. And you know that when sodium bicarbonate is added to water, it dissociates to produce carbon dioxide. And since carbon dioxide is the raw material for the process, it's going to speed up the process of oxygen evolution. It is evolving, but I don't know whether you can see or not. Okay, now see. This setup shows
you can shine
that
a light
oxygen
from
is
the other
evolved.
side.
Which side?
Excuse me,
That
ma'am,
side
my
or this
your
side?
video is freezing.
Yes.
Ma'am, from the opposite side. Ma'am, from which side we are watching from the opposite side if you shine a light? Yes, ma'am. From there.
No, I think
Ma'am,
you
it
can
might be
see the
visible.
bubbles. Just see whether you can see the bubbles or not inside the funnel. Can you see the bubbles inside the funnel?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am, minutely. Yes, ma'am.
Can you
Yes,
see the
ma'am,
bubbles?
minutely. Yes, ma'am.
Yeah, minute bubbles only, because I set it in the morning.
ma'am.
Okay, that means at around 9:30, 9:45 I set it. Okay, so what is this experiment all about? I have taken some twigs of hydrilla plant, and you know that hydrilla is a submerged hydrophyte. And for conducting such kind of experiments, we normally use submerged hydrophytes like hydrilla, like elodea, like vallisneria. So few twigs of hydrilla I've taken. And then what I did, I placed it inside this beaker containing water. We should have taken pond water. So normally pond water is taken because pond water contains more carbon dioxide than tap water. But here I have taken tap water. So you take few twigs of hydrilla, then you take a short stemmed funnel and you cover those twigs of hydrilla inside the water. Then you take a test tube, fill it with water up to the brim and carefully invert it over the stem of the funnel, which I have done. Then I placed it in sunlight. After placing it in sunlight, maybe after some time, I could see, bubbles evolving from the cut ends of the twig and, going to the upper part of the test tube. But now, and when I had added a pinch of sodium bicarb also in sunlight, what happened? The rate of evolution of bubbles, it increased. Okay, I saw that, but now since we are not getting ample sunlight, this is not showing exactly, but still bubbles are inside the funnel. Inside the funnel, it's all full of bubbles. Now, what questions will be asked from this part? So they are going to set this thing up. That means diagram-based question will be given, and you'll be asked to identify what is the aim of the experiment. So you'll write to show that or to demonstrate that oxygen is evolved during photosynthesis. Then what kind of plant is taken for this? You will write submerged hydrophytes. Give an example of such kind of plant. So you will write hydrilla or elodea. Then actually the funnel should have been placed on small blocks of wood. I couldn't get hold of that. So small blocks of wood so that it is slightly raised from the bottom. You know why? So that to allow free circulation of water and a continuous supply of carbon dioxide would be reaching the hydrilla twigs. Okay, because if the funnel is completely touching the bottom of the beaker, then the amount of carbon dioxide in that contained on that trapped water is confined. It is limited. So after that is exhausted, the process is going to stop. So therefore, the funnel should be slightly raised from the bottom. This is another question which is asked. Why is the funnel raised slightly above the slightly above from the bottom of the beaker? So you will write to allow free circulation of water so that a continuous supply of carbon dioxide is provided to the hydrilla twigs. Then next thing is what happens when a pinch of sodium bicarbonate is added? You will write the rate of evolution of oxygen increases because sodium bicarbonate dissolves in water to give out carbon dioxide. And since carbon dioxide is a raw material for the process, so the process, the rate of the process increases and the rate of evolution of oxygen increases. Are you clear with this experiment?
Yes, ma'am.
Okay. Now. Next. This is an experiment to demonstrate osmosis, basically endosmosis and exosmosis. So before coming to endosmosis and exosmosis, you must know what is osmosis. So what is osmosis? It is the movement of water molecules from a region of its higher concentration to a region of its lower concentration through a semi-permeable membrane. So two things you must remember to complete the definition of osmosis. First, the water molecules are moving from a region of their higher concentration to a region of their lower concentration. That means higher concentration of water to lower concentration of water. All right, that's the first thing. And second thing is that they need a semi-permeable membrane. A semi-permeable membrane is a membrane which allows only water molecules to pass through. All right, these two things are required for the process of osmosis to occur. So what I did, I had taken a potato, a raw potato, I peeled it and I cut it into strips. These strips, you can see the strips of potato which I have cut. Can you see the strips of potato? Yes or no?
Yes.
Yes, ma'am.
Yes, ma'am.
Okay, they were all of equal size. Okay, but what happened? One I placed in water, this one I had placed in water, and this one in concentrated sugar solution. Okay, I had placed it for few hours. You know what happened? If you can see this thing, the size has definitely increased and it is so very, very rigid. You cannot bend it also. It cannot bend. It will break, but it will not bend. You can see I'm trying to twist it. It is not bending. It is so very tough, okay? So hard and rigid. That means what has happened over here? Endosmosis has taken place. What is endosmosis? The inward movement of water molecules from a hypotonic solution to a cell whose concentration is more than the outside. What does that mean? That means the potato strips, they have cells. The cells, they have a cell sap. The cell sap has a higher concentration than the outside water. That means water is dilute, whereas the cell sap is concentrated. So water molecules are going to move from a region of their higher concentration. Their higher concentration means the water outside. When I'm saying that I have taken only water, that means 100% water I have taken. Water potential is maximum. So the water molecules are going to move from outside to the cell sap of the potato cells. And as a result, since it has accumulated water, the cells have all become very, very turgid and tough and rigid. And that is why this is so very firm and it is not bending. But on the other hand, the same size, this potato strip I had taken, but look at the condition of this potato strip, which was kept in strong sugar solution, okay? And you can see what is happening to it. Can you see that? Can you see what
Yes,
is
ma'am.
happening to it? What am I
Yes,
doing? It is all the strips. You can see that they've become absolutely flexible and I can bend them, right? So the same size of potato strips I had taken, but one that was the condition, it was kept in hypotonic solution, and this one I had kept in hypertonic solution. That means a solution whose concentration is more than the concentration of the cell sap. So when a cell is placed in such a solution, exosmosis is going to take place. That is, outward movement of water molecules is going to take place from the cell sap, from within the cell to outside. Since outside is concentrated and inside is dilute, water molecules are going to move from a region of their higher concentration to a region of their lower concentration. And as a result, this is the condition. So the potato strips have become flaccid and they are indeed flexible. You can see that, okay? They have lost their turgidity completely. So this experiment demonstrates endosmosis and exosmosis. Are you clear?
Yes.
Yes, ma'am.
Yes, sir.
Very good. The same experiment, I did it with potato osmoscope. This is referred to as a potato osmoscope. Okay, so what is, you can also make this with a carrot, and then it will be known as a carrot osmoscope. What is an osmoscope? Osmoscope is an apparatus to demonstrate the process of osmosis. Okay, osmoscope demonstrates the process of osmosis. So I took raw potato and I peeled it, then I cut it into half. And then what I did, I cut out the edges to flatten it so that the potato can sit inside these containers. Then I scooped a cavity. I made a cavity at the center of each of the potato piece so that it can hold some substance. So I scooped out a portion of the potato in order to create a cavity. And in one, I placed glucose powder, and another one I placed corn flour. Okay, and this third one, what I did, I had taken a boiled potato and I cut it into half, scooped the center, and I kept the glucose powder. All right, so three situations you are seeing. This is the first one, raw potato, half of the raw potato with a cavity at the center containing glucose powder, and this another half of the raw potato with a cavity at the center containing corn flour. And the third one is a boiled potato with a cavity at the center containing glucose powder. Now I had placed this yesterday. Now what happened after a few hours, you can, I don't know whether you can see this or not, it has formed a solution. Can you see that? Can you see that? It has formed a solution.
Yes, ma'am. Yes, ma'am.
Inside, it has formed a solution. And here,
Yes, are all these potatoes peeled off?
Yeah, potatoes are peeled off. You can see that. I have peeled them.
Yes, ma'am. Yes, ma'am. Okay, ma'am. Thank you.
I have peeled and have flattened the bottom, all right? So that it can sit in such a way that the fluid is not entering into the cavity. The level of the fluid is below the level of the cavity. Okay, that means the water level is not coming up to the cavity. Water is entering, but water is not entering by overflowing, but rather what you can see the condition over here. You can see the water level and you can see the level of the potato also. The potato is way above the water level. Yes or no?
Yes, ma'am.
Yes. All right. Now in the second one, you can see what has happened. You can see that it is as dry as bone. You can see that the corn flour, I kept it since yesterday, but it is still the same. All right. So what has happened over here? What has happened? Why this has formed a solution and why no solution is formed over here? And here also there is no solution formed. What for? And what for no solution is formed? What is the reason? Reason is here, same way, since it has been kept in a hypotonic solution, water is hypotonic to the cell sap of the potato cells. So as a result, what will happen? End osmosis will occur. Water molecules are going to move from outside to the inside of the cell. That means to the cell sap of the cell. That is, end osmosis will occur. And because end osmosis has taken place, that is why a solution has been formed. Okay. Someone has unmuted. Can you please mute yourself? Is it anyone among our students? Is it anyone among our students? Then? Sorry?
Ma'am, it's showing Reena Matthews and Shweta Shaw.
Yes, ma'am.
Okay, okay. So then what has happened over here? So it has formed a solution, right? So that shows that end osmosis has taken place, and since glucose is soluble in water, so it has formed a solution. Here what has happened? Why there is no solution in the cavity? The reason is that corn flour is insoluble in water. Corn flour is insoluble in water, and that is why the solution hasn't been created. Now, what is the purpose of taking this boiled potato with, with a cavity at the center and keeping the glucose powder? What is the purpose of taking a boiled potato and conducting the same experiment? Taking a boiled potato and showing this experiment confirms that it can only be done. This process is going to happen only when we are using a raw potato. It cannot happen in a boiled potato. The reason is that when we are boiling the potato, the cell membrane is losing its semi-permeability. And once the semi-permeability is lost, what will happen? There will be no osmosis. Because I told you, two very important factors are required for the process of osmosis to occur. One, that they should have a concentration difference. One should be concentrated, another one should be dilute. And second thing is that they should be separated by a semi-permeable membrane. That's what I said. So here, the second factor is missing. That is the semi-permeability of the cell membrane. Since the potato has been boiled, the cell membrane has lost its semi-permeability and hence no osmosis has taken place. Then what's the purpose of taking this setup? They'll ask you, what is the purpose of taking a boiled potato? Answer is, this acts as a control setup. Okay, a control setup in which if at least one factor is missing, then the experiment cannot be brought to conclusion. I hope you are clear.
Yes, ma'am.
Are you clear with this? Okay, good.
Excuse me, ma'am.
Yes.
Ma'am, can we take sugar in place of glucose?
Yeah, you can take, no problem. Powdered sugar you can take, that will be easier, convenient. You can do it at home, no problem. You try doing it today also. I did it at home and I'm showing it to you. So you can also very well do it. Now this experiment is also based on osmosis, that means endosmosis and exosmosis. But this is done with egg membrane. Now you remember the examples of egg membrane, do you remember the examples of egg membrane? I mean examples of semi-permeable membrane, do you remember?
Yes,
Yes,
What is a semi-permeable membrane? What is a semi-permeable membrane? I said a semi-permeable membrane is a membrane which allows only water molecules to pass through, right? So it can be cellophane paper, it can be parchment paper, it can be sheep's bladder, it can be goat's bladder, it can be egg membrane. Now, how to get this egg membrane? So what I did 4 days back, I took 2 eggs and I put them in vinegar. Okay, in what? In vinegar. Vinegar contains acetic acid. So the acetic acid reacts with the calcium carbonate of the eggshell and dissolved the eggshell completely. Are you getting what I said?
Yes,
Did you understand what I said? So the eggshell was completely dissolved, leaving behind the egg membrane only. The egg membrane was only there. Okay, have you ever seen the egg membrane after cracking one egg?
No, ma'am.
Never. Okay, so let me just bring it out, then you can have a feel of that. Can you see what I'm showing? Can you see the yolk inside also?
Yes, ma'am.
yes, ma'am.
Yes, ma'am.
Can you see that? So this is that means you can see the yolk and the other things, the albumin very clearly because it is just covered by a translucent egg membrane. Okay, then what I did after 4 days, after the eggs were completely devoid of their shell, I washed them with water and one I placed in concentrated sugar solution and the other I placed in water only. Can you see the size difference? Here, can you see the, okay. Just a moment. Can you see this?
Yes, ma'am.
Yes, ma'am.
But can you see the yolk over here? Like you saw the yolk over there?
No,
No,
ma'am.
ma'am.
Like you saw the yolk. You cannot see the yolk, you know, here. This one and this one. Which one appears to be bigger? Which one appears to be bigger? This one, isn't it?
Ma'am,
Isn't it?
without yolk.
Sorry?
Ma'am, without the yolk.
I'm asking you which one appears to be bigger? This one or this one?
Ma'am, the
Ma'am,
right.
that right.
And the one which are holding in right hand.
This one, isn't it?
Yes, ma'am.
This one, isn't it? Yes. So this is the one I had placed in water, and this is the one I had placed in sugar solution, concentrated sugar solution. And what happened after that when I placed them in two different kinds of solutions, one in water, one in sugar solution? Obviously, the one I placed in water showed end osmosis, and the one I placed in sugar solution showed exosmosis. So since water entered into the cells, so that is why it led to the swelling up of the cell. And here, since water left the cell, that is why it led to the shrinkage of the cell. Okay. I hope you are clear now.
Yes,
Yes, ma'am.
All the experiments are clear on osmosis, on endosmosis, exosmosis with a potato osmoscope with potato strips, because this potato strips, you have this in the exercise question. Osmoscope, you might be asked. You are going to come across this question in the test papers regarding osmoscope. You can have a potato osmoscope or with a carrot osmoscope, the same process. And the hydrilla experiment and the photometer experiment to show that rate of transpiration. That's it. Any doubt if you have, you can ask me. Otherwise, we can wind up. Any doubt?
No, ma'am.
No, ma'am.
Absolutely clear then. So then most of the experiment basing on absorption by roots and transpiration and photosynthesis, only one I could show, you have seen. All right. So total, how many experiments did you see? First class I showed 4, and today I showed 5, isn't it? 5 experiments.
Yes, ma'am.
Yes, ma'am.
Okay. Okay. Thank you so much. Thank you for attending the class. Bye-bye.
Thank you, ma'am.
Thank you,
Thank you,
ma'am.
ma'am.
Thank you, ma'am.
Bye-bye.
Thank
Bye-bye.
you.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Yeah. Thank you. Thank you.
Yes, ma'am.
Leave silently. Okay. Thank you, children. Leave. Ayush, Arnav, Dev, Varun, please leave. So some of you are there. Arnav. Abhishek, Pratyush, Rahul Chaudhary, Nandini, Ipsita. You may leave. Varun. You're still there. Arnav, Dev. That means you have logged in, you aren't there. Is it like that? Dev Udaypuria, are you there?
The person you are calling.
Arnav Dasgupta, Dev Udaypuria, you may leave. Ipsita, Rahul Chaudhary, Shobik Mukherjee, Abhishek Das. Dev, what are you doing? You may leave, I said. I've been telling you for the past 5 minutes.